It's a pleasure to introduce today's annual Arthur W. Ludwig Lectureship in Humanism and Medicine. This lectureship is part of our Advancing Idealism in Medicine initiative. It's a resident-founded program that was started over a decade ago to promote trainee well-being and includes, amongst other things, this lectureship. We have a Grand Rounds lectureship as part of AIM to bring in inspirational role models in the practice of medicine who we can all learn by their example. And that is certainly true for today's speaker. But first, let me tell you a word about Arthur Ludwig, for whom the lectureship is named. Dr. Ludwig arrived at Mount Sinai in 1943 as an intern, and he remained affiliated with the hospital throughout his entire career. He completed his internal medicine residency and followed with fellowships in both endocrinology and pathology. He served as an attending in the endocrinology clinic and was chief of what was then the outpatient department's diagnostic clinic. He was a true clinician investigator. He combined scholarly research with a very active patient panel and practice. And he was regarded by his peers as a marvelous diagnostician who was really a strong patient advocate, dedicated to providing superb and compassionate care for which his patients really were dedicated and he earned their highest level of trust. Today's award lecture goes to Dr. Marc LaForce, who's currently the Director of Technical Services at the Serum Institute of India, and he's also clinical professor of medicine at the NYU School of Medicine here in New York. Dr. LaForce completed his internal medicine and infectious disease training in the Harvard system up at Boston City Hospital and was influenced by an early post with the Epidemiologic Intelligence Service of the CDC. He went on to hold tenured academic positions at the University of Colorado School of Medicine and the University of Rochester School of Medicine before shifting gears altogether later in his career and committing himself wholly to global health, which was a bold and courageous move that literally has benefited millions of people. From 2001 to 2012, he served as director of the Meningitis Vaccine Project, which was a partnership between the World Health Organization and PATH, which developed, tested, and led in 2010 to the introduction of a new and affordable Group A meningococcal vaccine for use in Sub-Saharan Africa. This vaccine has now literally been administered to millions of people thanks to Dr. LaForce's efforts. And so we're delighted to present him this morning with the Arthur Ludwig Award. Dr. LaForce. Congratulations, and he'll get this nice plaque at the end. Welcome.
Thank you. Thank you. It's a pleasure to be here. And what I'd like to do this morning is tell you a story and reflect a bit on what that story has told us. The story is really focused around a bad disease and a major problem, and that was epidemic meningitis in Sub-Saharan Africa. About 90% of the global burden, you remember this now, the global burden of meningitis is clustered in this belt. And this is what's called the meningitis belt in Sub-Saharan Africa. And it's a difficult area, and we're going to talk a fair amount about this country, Burkina Faso, which is one of the hyper-endemic countries that's located exactly in the belt itself. Some of the characteristics we'll talk about, there are major epidemics of meningitis in this area until recently that occurred about every 10 to 15 years that were frankly quite harrowing. This area, I don't know if any of you have traveled to Sub-Saharan Africa, but if you have, there's a collection, these are the poorest countries in the world. They have few natural resources. The climate is, I think I'm generous in saying it's inhospitable. It's hot, dusty, it is a difficult climate. Most of the people work in rural areas and the per capita income in these countries works out to be about $1, $1.50 a day. So that an annual income for a family would be somewhere in the range of $400 or $500. They have no disposable income. This is high noon in Ouagadougou, Burkina Faso. And this cloud of dust is called the Harmattan. And the Harmattan, because of wind changes in Sub-Saharan Africa, the winds, instead of coming from the Gulf, that is moving south to north, switch around and then start blowing from north to south. And as they blow from the north to the south, they actually collect all of this dust from the Sahara Desert. And so what happens is in the January, February, March, and April, there is this constant cloud. And it is felt that this has a lot to do with determining epidemics of meningitis, because epidemics of meningitis only occur during that time frame, that is from January to April or May each year. And this is what a meningitis outbreak in a place like Burkina Faso, well, this is a Burkina Faso district outbreak that I helped work up in 2006. And these are weeks of the year in 2006. And again, you see that from weeks 1 through 8 or 9, there's really not much going on. And then all of a sudden, there are hundreds of cases of acute meningitis that are occurring in a population of about 134,000 individuals. This lasts for about 1 month. And when we finished the tally, there were 1,000 cases of meningitis that occurred over a month's time in a population of 134,000. I ask you to think, Upper East Side, I've looked it up in terms of the census tracts. Your population here is about 230,000 for the Upper East Side. Just reflect, what do you think would happen if you had 1,000 cases of acute meningitis that occurred over a month's period of time in the Upper East Side? Well, if you just sort of let your mind sort of jump a little bit, that's exactly what happens in this particular area. Everybody stays at home. There is no commerce. Everybody just sort of hunkers down until these episodes are over. And so it is really hard to find food. It's hard to get to water. People are literally afraid of coming into contact with anyone else during these particular episodes. And when you put it together, the 3 countries that are there, we have Burkina Faso, and then on either side of it, Mali and Niger. And if you look over a period of time from 1986 to, this goes out to 2008, all of these peaks are peaks of meningitis. And I call your attention to the ordinate on this particular graph. That's 5,000. So in Burkina Faso, in 1996, and we'll come back to this epidemic a little bit later on, they were having 5,000 cases in a week. So these are extraordinarily severe events that completely change not only the demographics, it just changes society. And when you add it up, epidemic years here are summarized for Burkina Faso, Mali, and Niger, and you can see for yourself the numbers These are district incidents per 100,000 of 158. And you say, well, how do I put this into context? Just remember that in the United States, our rates are 0.3 to 0.5 per 100,000. So we're talking about incidence rates that are 100 times, easily 100 times higher in West Africa. And furthermore, when you actually drill down and you go down into communities, I've been in communities where the incidence rates for that particular community have been 2%. So when we finish, tallying up all of the cases and counting the denominators within those particular populations, 2% of the individuals residing in that are ill from meningitis, all occurring over a month and a half. So it's not a fun disease. And because there are no doctors out in the periphery, when the epidemics come, and you can never predict which part of the country the epidemics are actually going to start, the hospitals are generated where people fall. So people then organize themselves and then bring the sick to areas that they just basically create lean-tos, and these are how individuals end up getting treated. This was a youngster, again from the Busset outbreak, this youngster just had a grand mal seizure, and he's being cooled down, from water out of this particular jug, to give you an idea in terms of how difficult some of these areas are. And I also wanted to remind individuals that it's not just the mortality from meningitis, but it's what's left behind. If you have meningitis, at least, and are not really taken care of immediately, you're never as smart after the episode of meningitis that you were when you went into it. This is a young boy who has survived his episode of meningitis, but he's stone deaf. And the commonest cause of acquired deafness in Africa is survival from meningitis. And being deaf in Africa is not a good formula for survival. And when you put all of these data together, these are WHO data from 1950, and I use this because I think it's important to have a long-range view of this. And you have, again, these major epidemics that occur about every 10 years. And if you start adding them up mentally, and this is just cases reported, that's not necessarily all of the disease that's occurred, you quickly get to around 1 million cases. This is, again, the size of this particular issue. And the disease really relates to an organism that for most of us here in the United States is a pretty quiescent organism, Neisseria meningitidis. It resides in the posterior nasopharynx. It really is pretty quiet. It's all of us get infected at some time during our lives, and the organism is carried for a while. There's an immune response, you clear carriage, and then that's the sort of the end of it. And the organism basically sort of circulates, and again, for reasons that are not very clear but are really pretty dramatic in Africa, the organisms actually spill into the bloodstream, causing these episodes of meningitis and very severe toxemia. What to do about this? Well, clearly the most sensible strategy, if you're going to try to deal with a problem like this, is to prevent it. Trying to treat an episode, we were talking about this at dinner last night, trying to treat episodes are really, I view from a public health standpoint, are really just trade-offs. It really is sort of second class. What you really want to do is you want to make sure that something never happens. And scientists, and particularly Emil Gottschalk, those of you may know Dr. Gottschalk from Rockefeller, in the mid-60s, brilliant work at Rockefeller was done particularly in terms of the capsule that surrounds Neisseria meningitidis, and the studies that were done by Dr. Gottschalk's group and the Army Institute of Research, showing that the capsular material was antigenic and that antibodies against that capsular material were preventive or could be or could prevent disease. And the flow of product really began with polysaccharide vaccines, but soon went to conjugate vaccines. And by 2005, in the United States, there was a polyvalent conjugate vaccine that was available. Now, for those of you who aren't aficionados of vaccines, you're going to say, well, what difference does it make between a polysaccharide vaccine or a conjugate vaccine? And it turns out it's a very, very important distinction. A polysaccharide vaccine is basically stripping off the capsule off a particular organism and then using that as an antigen. The problem with it is, from an immunologic standpoint, it only stimulates B cells. And so you can have a decent immune response, but that immune response is usually short-lived, gone within 2 to 3 years, without memory. But if you take the sugar capsule and you combine it with a carrier protein, you create a new vaccine called a conjugate vaccine. It's simply taking the capsular material and annealing it to a carrier protein. And when you do that, the conjugate vaccine really is a much, much better product. It now works in infants that are under 24 months of age. The avidity of the antibody is high. There is memory that's induced, so that if an individual sees the same antigen 5 or 10 years later, it mounts an anamnestic response to that particular antigen. And importantly, it has an effect on colonization. If you're immunized with a conjugate vaccine, this perturbs this relationship of carriage such that meninges tend to get cleared from the nasopharynx post-dosing with a conjugate vaccine. So there are major advantages in terms of meningococcal conjugate vaccines. Well, what was the situation in terms of vaccines that were available in 2001? Well, in 2001, there were largely only polysaccharide vaccines that were available. And these were largely used in what are called, reactive vaccination campaigns. And reactive vaccination campaigns are defined that when epidemics occur, there usually is an appeal for vaccines and for funds. These go through WHO, UNICEF, et cetera, and donors are identified, vaccine manufacturers are identified, and these vaccines get shipped to these particular countries that are having the epidemics, and reactive vaccinations are done in those particular areas. Well, given the length of time it told me to describe this, you can imagine it takes time. And so we already went through this exercise that an acute meningitis epidemic is over in a month. And these particular reactive campaigns were, from a public health standpoint, were really generally ineffective, but politically very necessary because governments have to do something. And more importantly, there were no plans from any of the major vaccine developers to develop a Men-A vaccine for Africa, largely because there's no Group A meningococcal disease in the United States, or in Europe for that matter. The only place in Europe would be Russia, and they have their own vaccine issues. And that is what led to the development of the meningitis vaccine project. And this is, with all of this as a preamble, is the beginning of really the story. In 1996, after about 500,000 cases of, not 500,000, I'm sorry, 250,000 cases of meningitis in Africa, the ministers of health from African countries got together, went to WHO and basically said, please help us. We got to do something about this. And under WHO leadership, there were a series of international meetings that led to a strong recommendation that there needed to be a development of a Group A conjugate vaccine. This led to a partnership that unfolded between PATH, which is an NGO in Seattle, and the World Health Organization. There was a grant submission to the Gates Foundation, and the Gates Foundation in 2001 awarded PATH and WHO a $70 million grant that was a very unusual grant. The money was given up front, and it was established as a 10-year grant with a single goal. And that goal was to eliminate epidemic meningitis as a public health problem through the development, licensure, and introduction of a conjugate meningococcal vaccines. That was in, that would have been in May of 2001. I was asked to sort of lead the effort in June, and one of the first things that one does is sort of look and review the information that are with all of the meetings and the stuff that took place. And what was missing, I thought, as well as WHO people, was really African input. There were lots and lots of pronouncements. Meetings took place in Washington, Seattle, and Geneva were the three areas. But there were no meetings in Nairobi, there were no meetings in Ouagadougou, there were no meetings in Dakar, and there really wasn't very much input in terms of the customer, that is, the African that was part of this. And so we spent the first 9 months in Africa basically talking with ministers of health, ministers of finance, EPI directors, infectious disease clinicians, et cetera. And we soon learned that cost of vaccine was probably the most important limiting factor to the introduction of any new product in Africa. And furthermore, in discussions, particularly with ministers of finance, we said, well, okay, what do you mean by cost? In other words, what would work? And they said, what would work, again, never any guarantees in Africa, but a vaccine would have to cost somewhere in the range of 50 cents a dose for us to be able to sort of work that into our current budget plans for vaccines within that particular country. Now, at that time, this would have been in the fall of 2001, spring of 2002. U.S. conjugate vaccines that had meninge A were costing between $130 and $150 per dose. And so that's a pretty big difference between 50 cents and $150. So that was the first major challenge that came. It was, number one, finding out what is a price point that would help guarantee success in Africa. And that turned out to be very, very important as time went on. The Gates Foundation felt that when the $70 million was given to us, that we would have discussions with major vaccine manufacturers, we'd cut a deal with one of the major vaccine manufacturers, transfer funds to them, and then let them do it. And so we began to have discussions. And as you can imagine, the vaccine price was an important issue because for this to work, our point was we're talking about something significantly less than $1 a dose. Pharma was interested in ensuring that there was guaranteed purchase and that there would be major investments in manufacturing capability for them, and essentially creating a no-risk model where in the end, basically they would decide what the final cost of that particular vaccine was. And so in the spring or late spring of 2002, we realized that we couldn't reach an agreement following that particular strategy. So we decided to become our own company. With the $70 million in the bank, we decided to become a virtual vaccine company and that we'd develop our own Group A conjugate vaccine. There were two elements in there that were crucial in making that decision. Number one were the inputs from the Africans, and two was a business plan that had been commissioned by WHO that indicated that the cost of goods for making a monovalent Group A conjugate vaccine could be as low as 20 cents a dose if one were talking about making somewhere in the range between 25 and 50 million doses of vaccine per year. The volume issues we'll get into, but those two elements were really important in terms of the decisions that we then made to pursue making our own vaccine. And so over the next several years, we put together a team because we needed raw materials. Number one, there were two key raw materials. One was vaccine-grade polysaccharide, the sugar, and two was a carrier protein, and we chose tetanus toxoid as the carrier protein. The Meningitis polysaccharide was made by a company in Amsterdam called Cinco, and we did a global search for a vaccine manufacturer that would be willing to sort of work within the confines that we had described. And I spent the better part of 18 months or 2 years, literally, doing very specific analyses of vaccine companies in Brazil, India, Indonesia, Mexico, with dossiers, and then finally we put all of this information together and we chose the Serum Institute of India in Pune, India as a vaccine manufacturing partner for a variety of reasons. Perhaps the most important is that it had already licensed 10 or so vaccines with WHO for international use. But we were really stuck with a conjugation method. To actually take the polysaccharide and combine it with a carrier protein requires a chemical reaction. And those chemical reactions, as you can imagine, are heavily protected by patents because the pharmaceutical manufacturers had already made these particular vaccines. And so there was a thicket of IP issues and patents protecting actually making this particular vaccine. And it didn't seem to make any difference when we had conversations with these vaccine manufacturers in terms of saying, look, this isn't competitive. There's no MENA in the United States. It's specifically for Africa. The answers were no. And so we were really stuck. And it is here where we ran into what I call the best of the United States. And the best of the United States was a wonderful collaboration that evolved with the Food and Drug Administration. At that time, the Food and Drug, and some of you may know Carl Frasch, who was the head of the vaccine unit for many, many years. And Carl and I have known each other for a long time. And as we worked through our problems in terms of trying to identify something, he quietly told me, he said, look, Mark, if you're sort of stuck, he said, why don't you think about coming and paying us a visit? We may have something for you. And so in the, it would have been the summer of 2003, after a meeting in Geneva, I visited Carl and met a brilliant scientist, Dr. Robert Lee. And Robert Lee had designed and evolved a whole new conjugation method. And to make a long story short, after discussions at FDA, they were willing to transfer the IP and all of this to us for essentially nothing. And so this intellectual property was then transferred to PATH. PATH then did a sub-license with Serum Institute of India. And the method was really quite interesting. The group A polysaccharide is a long polysaccharide, and chemically, to actually get this annealing between a carrier protein and the polysaccharide itself requires what's called activation with periodic acid, such that there are diol groups that are transferred to become really aldehyde groups that are extremely reactive. Those aldehyde groups then can react with amino groups that are on the carrier proteins, and then they serve as a clamp. And so this entire process had been developed with Robert Lee, but it had one important element, is Robert had worked out a way to activate the carrier protein as well as the polysaccharide. And so what happened is the yields that you received were vastly better. If you're making a vaccine that you're going to sell for $150 a dose, it really doesn't make much difference what the yields are, because it just means you just have more raw materials, but the profitability is so big on the other side, it doesn't make it a difference. But if you're looking at a vaccine for 50 cents a dose, efficiency becomes crucial in terms of being able to keep cost of goods low enough to be able to make that possible. And that's what this genius at FDA was able to provide, such that the final conjugate, these are representatives of tetanus toxoid, and these are these arms of the polysaccharide that actually basically stick out, and it turned out that this particular formulation was the most immunogenic formulation for a group A conjugate vaccine that had ever been tested. And again, when we say, you always talk about the government being so slow and, nothing ever happens, et cetera. I had my first meeting with them, it would have been first week in August in 2003. And by December of 2003, over the Christmas holidays, the first group of Indian scientists were working at Carl Frasch's lab to actually learn this particular technique. This was really warp speed. And after a lot of work, this is the vaccine as it stands. It carries the name Menafrivac, and it's a Men-A conjugate vaccine with 10 micrograms of polysaccharide, 33 micrograms of tetanus toxoid. And then we tested this in an international collaboration in a series of clinical trials. Any of you who've done clinical trials know that the rules for these are very stringent, and they should be. These are not funny issues. This just takes a lot of time and a lot of effort. The sponsors, there were two sponsors, obviously the Meningitis Vaccine Project, and the second sponsor was the Serum Institute of India in Pune, India. We had two CROs, and we wanted to invest in terms of CROs so that when we finished our work, we left CROs in those areas to be able to do more work. So we had a CRO in Dakar and a CRO in Mumbai. These were all the clinical sites, obviously in West Africa and in India, and the laboratories that were used to actually measure all of the results were really global. CDC in Atlanta, the CBER labs at the FDA, the Portland labs or PHE in the UK, the Max Planck Institute. This was really a global effort in terms of categorizing the laboratory parameters of this particular vaccine. And I'll show one slide of this, but just to sort of show you, this is an aggregate of the data in terms of the West African studies, 12 to 23-month-olds, 2 to 10-year-olds, 11 to 17-year-olds, and 18 to 29-year-olds. And this is comparing the conjugate vaccine with a polysaccharide vaccine. This is 0, and then this is 28 days after immunization. And the point being is that at each age group, receipt of the Meningitis conjugate vaccine resulted in an immune response that was much better than seen with polysaccharide vaccines. Also, the data were very clear that the vaccine was safe. We felt quite confident as we moved into the introduction of this particular vaccine, and WHO agreed with us. It was licensed by the government of India, the Drugs Controller General, in 2009, and pre-qualification was awarded by WHO in June of 2010. And after major safety studies were done, we launched the vaccine. And this is one of our great friends. This is Blaise Compaoré, His Excellency, who was the president of Burkina Faso. As we worked through getting this vaccine ready for Africa, it became clear that the African culture is stuff that they like having a major person that usually is, in the French parlance, le patron. And the patron who agreed to serve in that particular function was Blaise Compaoré, the president of Burkina Faso. So he was essentially our front man in terms of talking with WHO AFRO, with other ministers of health, so that when the president of Burkina Faso calls, it's not like me calling. You know, they never answer me, but they always answered when Blaise Compaoré called. And so we introduced that vaccine in December of 2010, and it was one of the most exciting days in my entire life. Picture a large horseshoe arena with the speakers and it wasn't the speakers were sort of on the inside, but with about 5,000 kids that ranged from, I don't know, 4 years to about 12 years of age, parents, dignitaries from WHO, UNICEF, Gavi, et cetera, that were there. And Blaise Compaoré addressed the group in Bambara and also for all the international guests in French. And I'm fluent in French, so I had the privilege of being able to listen to exactly what he was telling his Burkinabés. And he basically said, we have an opportunity that may present itself only once to get rid of a particular disease. And he was calling upon all Burkinabés to participate in this particular effort. And he said, to show participation, he said, we have all of the kids that are here, and you could have heard a pin drop. The kids were absolutely mute as this was going on. And basically saying, he addressed to the kids in terms of saying, you must be vaccinated before you go home. and you must show your parents your vaccination certificate. We have set up vaccination groups in every single school in all of Ouagadougou, and we've set up an army of vaccinators at the end of the horseshoe. But whether you get vaccinated there or whether you get vaccinated in your local school, you have one responsibility today, and it's a responsibility to your country and to your president. Woo, this is really. And then what unfolded over the next 10 days was the most amazing vaccination campaign. I stayed through the whole campaign. These were thousands and thousands of individuals who presented themselves to be immunized. The target population were all 1 to 29-year-olds were targeted to get a dose of this particular vaccine. The target population was 10,677,000 Burkinabés who fell into this age group. The campaign lasted 10 days. They were finished by day 9. And they immunized 10.8 million persons. That's about the population of New York City, right? Good luck. It was a highly successful campaign, as you can imagine. And these are the EPI data for Burkina Faso. These were the 2000, we'll come back to this, 6, 7, and 8 Meningitis epidemic. And the vaccination campaign occurred in 2010. We were very concerned to make absolutely sure that we had the best possible surveillance data for this particular introduction, because this was really going to set the tone for everything. And in that year, that is the 2011 epidemic year, there were 3,800 suspected cases of meningitis. And of that, 3,400 spinal taps were done. These were done in all health clinics throughout Burkina Faso, and they were done by nurses. Nurses are trained, they do the LPs. Doctors don't know how to do LPs, but nurses know how to do LPs. And what we had in terms of our surveillance base, the number of CSF samples that were sent for lab confirmation was 97% of all suspected cases of meningitis that year ended up being tested. A third were positive, which is exactly what we have in the United States. In the United States, we're at 35% in terms of suspected cases of meningitis with positive isolates. And we found one case of Men-A disease that particular year, and this was in a young woman who came from a desert area between Mali and Burkina Faso that had not been immunized. And when we put these data together, these are cases of reported cases of meningitis, and these are the fractions of CSFs that are obtained that yield Group A Neisseria meningitidis. So if you look at this outbreak for 2006, 7, and 8, you have about 45,000 cases, 85%, 91%, 80% of all CSFs that year yielded Group A disease. The vaccine was given, went from 25% to essentially 0 in 1 year. Very dramatic result. And we tried to understand what was taking place. And at the same time, we were doing a carriage study with the Norwegian Institute of Public Health. And this was a carriage study in a couple of rural areas and 1 urban areas. These were beautifully designed studies. The Norwegians are really, and it was partnered with CDC. Both the Norwegians and CDC really know how to do this work very, very well. And I won't belabor much of this, but this is before vaccinations were done. And out of a total of 20,000 throat cultures that were done, we were able to establish a carrier rate for Group A at less than 1%. It was at about 0.4% carrier rates in these 3 areas. Now, there were big differences between the areas, but they averaged out to that. Post-vaccination, in 5 separate studies, a total of another 22,000 throat cultures that were analyzed both locally and then all sent to Oslo for verification. Not a single group A was identified post-immunization. These data were incredible because they were consistent with vaccine-derived herd immunity with disappearance of disease, but also absence in terms of group A carriage. And we wondered, well, you know, are we doing something wrong? Because the results were actually better than we had anticipated. And we concluded that no, that there was a powerful effect because the vaccine was a very powerful vaccine, and the coverage within the vaccination plans had been so intense. We also reflected on what was happening to an element in epidemiology that's called the basic reproductive rate. This is a very important concept if we're talking about disease where organisms are really carried in the posterior nasopharynx. The basic reproductive rate, it defines the transmission potential of a particular organism, and it's defined really as R0. And it's dependent on a simple ratio of the contacts per unit time times the transmission rate per contact times the duration of infectiousness. Well, the study results, we knew that the vaccine itself seemed to block colonization, so that we felt that P certainly would fall in this particular ratio. And we also felt that it shortened carriage, so that that would result in D being decreased, so that the overall impact would be a fall in R0. The interesting thing about R0 is when R0 falls to less than 1, the agent disappears from the population. It no longer has the ability to last in the population. And all of these data were consistent with what we had observed with the introduction of this particular vaccine. And what we think happens is this. This is back to the cartoon that we had before with the circulation of this particular organism. And what we think happens is that you now have set up a series of walls so that when the organism is coughed onto somebody who has already been vaccinated, that individual is now fully protected in terms of being colonized, not only ill, but colonized with that particular organism. Well, then, from over the next 7 years was a rollout campaign. This was from Burkina Faso, Mali, and Niger is when we started, and then all of this was like a balloon. It just expanded over Sub-Saharan Africa. And such that as of now, we have 280 million Africans have been immunized. The number of Group A cases that were reported in a population of 350 million in 2017 was 2. 2 cases that were identified in the entire subcontinent. Again, testimony to how powerful this herd immunity, once it's achieved, it really controls disease in ways that we didn't quite understand at the beginning of all of this. And then we generated a sort of a report card, because people were sort of asking us, well, how much did it cost you, for all of this stuff? Developing and testing of the vaccine in round terms worked out to be about $100 million. That is 1/10 of what is cited as the usual cost for the development of a new vaccine. This is the first new vaccine to be internationally licensed, not made in pharma. So it cost about $100 million. The mass vaccination programs cost about $400 million. We had to maintain EPI coverage now. This has to be introduced into the expanded program on immunization. And these were the costs. In terms of the savings, they were now huge savings. The savings for families. We had excellent data that showed that in Sub-Saharan Africa, if you had a case of meningitis, that case of meningitis cost your family $100 is what it cost the family. It would be a rounding error here in the United States, but it's $100, but $100 represents 3 months of disposable income for a family. And the more we studied this, the more we realized that episodes of meningitis are important causes of poverty. Because what you do is you take out 3 months, and it just sort of cycles a family into a lower level. And so eliminating all of that, it really points out a major savings. There were major clinical savings also in terms of the 20% or so of survivors that are maimed that must be maintained by their individual families. And in the end, in the end, it works out that it cost about $100 million to prevent 1 million cases of meningitis and 200,000 disabilities. And I would submit that's an unbelievable bargain in terms of being able to do this. Okay, what are the remaining problems? Life is always a bit complicated. Men-A is now gone, but it turns out that represented 80% of the problem. You're still with 20% of the problem. And when you take away 80% of the problem, that 20%, believe it or not, becomes 100%. And so what happened all of a sudden is it wasn't thank you, it was, whoa, wait a second, what are you going to do with these? And so and also taking care of Men-A and also improved surveillance got us really now looking quite carefully at what else was going on. And what else was going on with Group C epidemics in Nigeria, Group X epidemics in Niger, and again, meeting with Africans, they basically said, well, how about doing this for an affordable polyvalent meninge conjugate vaccine? I won't spend very much time. This is a project that we're working hard in terms of being able to get all of this done. And let me close by saying that the African challenges over the next 10 years is make sure that Men-A continues to be introduced as an EPI, an expanded program on immunization, antigen, strong case-based surveillance, supporting, again, WHO regional work, and accessing introduction strategies for new polyvalent vaccines that will be introduced, hopefully, over the next 3 or 4 years. None of this occurs really with just a group. These are all the friends that we worked with throughout all of this. ANP, Cinco, PATH, CDC, et cetera. These are all exercises in collaborations, and these collaborations are absolutely crucial. And this was the team. We met only once as an entire group in Pune in 2010. But this was the entire group, the chemists, epidemiologists, vaccinators. This is the entire group in Pune at meetings that were the culmination of the development phase of the project. My thoughts are that I think with a polyvalent meninge conjugate vaccine and also more affordable pneumococcal conjugate vaccines, Africa can end up being the first meningitis-free continent by 2030. And that's the goal. The goal is to have a meningitis-free continent by 2030. And this was our starting point. Anybody who visited the office, we had a huge picture of this young boy. And this young boy was the customer. There was no other customer except him. Thank you.
Thank you so much. That was wonderful.
Is there any reason why that one belt disease was, but the first being relatively free?
Is there, yeah, the reason it appears to be a climactic reason. Again, this is the dust area and the dry area. And every single epidemic of meninge stops when the relative humidity gets up a certain level. So that it just, nothing works if it's wet. So it has to be dusty and dry. And those are the two characteristics.
Congratulations on your commitment and your success. There are 5 of us in this audience who are old enough to have had Dr. Ludwig as our teacher, and I'm one of them. I think he might say, if he had a voice here, number one, why are the CEOs of the vaccine companies not willing to make the vaccines available at cost or low cost? And the accompanying question might be, the belt that you showed in the original map of Africa seems to be a belt because of contiguous living conditions, but could be a genetic belt. What do we know about the genetics controlling these epidemics?
To answer your second question, the genetics has been looked at. And there is no unique risk factor to any genetic group within that particular area. In point of fact, there's plenty of disease in colonials who went to work in these particular areas. It appeared to be more climactic than it was genetic. The second, pharmaceutical companies, they're companies to make money, and there's no money to be made there, frankly. Now, how they make their particular decisions, I can't comment on that at all. But I do think that there is a point, though, where everybody has to say time out. Because I think letting a situation like this go on for as long as it went on is really unacceptable. And I think that is probably one of the most useful lessons of this, is that it doesn't have to be that way.
Did the numbers of meningitis cases decrease after implementation of the vaccine?
Yes.
Yes.
Hugely.
Thank you for a wonderful lecture. The immunologic response to this vaccine looks very robust. A lot of questions are about duration of immunity. Polysaccharide vaccines typically only a couple of years. What do we think about typical duration for a conjugate vaccine?
Good question and an important one. The best that we have right now is the surrogate of protection is the SBA, is the serum bactericidal activity. And so when we take the current SBAs, and we've gone out now 7 years, and we've followed individuals out 7 years, and if you plot out, over log T over a period of time, it looks like it would cross a titer of 1 to 128, which appears to be the most conservative estimate at what you could be at somewhere around 16 years. So it is felt that if this vaccine works, again, in a public health sense over this period of time, you're essentially talking about, you know, a single dose of vaccine protecting you for life for 40 cents.
So we're run out of time, but
Oof, I'm sorry.
that's all right. I want to really thank Dr. LaForce, and also I'd like to recognize and appreciate all, our gratitude to Mrs. Ludwig for sponsoring this course every year. Thank you so much.